So hello, everybody. First of all, I'd like to thank Montserrat and everyone from the Journal of Physiology for organizing this amazing two-day meeting. It's the first time that we do something like this, and I'm very excited. So thank you very much. And also thank you, Laszlo, for chairing the session. So today, my presentation is going to talk about molecular basis of impaired muscle function in a mouse model of a congenital myopathy. So first of all, let me point out my talk will be divided into two main sections. First, I'll give a short background introduction about congenital myopathies and myanogen receptor mutations. And please note that at the bottom of my slides or on the side, whenever you see the font change to blue, those are references that I'm giving. So if you want to know where I'm getting some of the information from, it's from these references. So first of all, let's look at the epidemiology of neuromuscular disorders caused by myanogen receptor mutations. Well, these as a whole occur approximately in 1 in 25,000, but most likely the prevalence is higher. And specifically, the most common congenital myopathy caused by myanogen receptor mutations is central core disease, followed by multi-mini core disease and central nuclear myopathy. As a whole, out of all the congenital myopathies, approximately 30% are caused by myanogen receptor mutations. And interestingly, 1 in 3,000 individuals carry myanogen receptor 1 mutations. So, as you might know, there's different forms of congenital myopathies that are caused, or different muscle disorders that are caused by myanogen receptor mutations. You have gain-of-function mutations, which are linked either to the pharmacogenetic disorder, malignant hyperthermia, and these are a gain-of-function mutation. Similar also, exertional heat stroke, rhabdomyolysis are caused by dominant myanogen receptor mutations. And these usually are mutations that cause the channels to become hypersensitive, so they open more easily at lower agonist concentration or lower activation voltages. Then you have those myanogen receptor mutations that lead to congenital myopathies. So these are different because the patients actually see a neurologist because they have a weak muscle phenotypes. And here you have different forms of disease. You have the dominant congenital myopathies, which are caused by dominant myanogen receptor 1 mutations. And the mutations could either cause the channel to become leaky or to become uncoupled. That is, when the DHP receptor gives a signal, the myanogen receptor doesn't open, so it doesn't transport calcium. Then you have those other disorders, which are due to recessively inherited mutations, which the most common is multi-mini core disease, central nuclear myopathy, and congenital fiber type disproportions. So this is what we call the class 4 mutations. They're recessively inherited. And one of the common things that is noted is that the patients have a decreased amount of myanogen receptor protein in their muscle biopsy. So if you do a Western blot, you'll see that there's less myanogen receptor present. So today I'm going to talk about group 4, that is, what's phenotypically characteristics of those patients with recessive myanogen receptor mutations. So first of all, they're born weak. So they could be either floppy infants, they have very weak proximal muscles at birth. They could also present with skeletal deformities. They could have a club foot, they could have scoliosis, and they often have feeding difficulties. Then an interesting finding in some of them, and most of those that have one allele with a nonsense or stop codon mutation, they have ophthalmoplegia and or ptosis, that is weakness of the eye muscles, as well as weakness of the facial muscles. Then they have the presence of multiple mini cores and or centralized nuclei on muscle biopsy. They have reduced levels of myanogen receptor protein. And interestingly, they have an increased level of transcript and protein expression of chromatin modifying enzymes, which we call epigenetic enzymes, which are the histone deacetylases, class 2, and DNA methyltransferases, Also, they have alterations in the microRNA expression. So what did we do? We decided to make a mouse model based on the 2 mutations that were identified in a severely affected child. So this child harbored a mutation in exon 36 that led to a premature stop codon. So it was a frameshift mutation. And he inherited the other mutation from his asymptomatic father, who carried a missense mutation in exon 91. This child was severely affected. He never achieved walking or sitting, and he actually sadly died when he was a baby. So this is, let's call it the Basel mouse model. So the Basel mouse model was made by actually Pavel Pegsar, who's the head of a transgenic mouse facility in Basel. On the left of the slides, you could see the genomic DNA of the constructs of the mouse constructs and the injected mice that were made using the CRISPR-Cas9 technique. So you see here that several exon 36 mutant mice were made, and they were germline specific. And on the bottom panels, it shows the PCR amplified genomic DNA that was used for the genotyping. So this is from exon 36, exon 91, and he inserted a PVU2 site so we can easily identify the wild type or the mutant mice. So as I said before, the exon 36 frameshift mutation was inherited from the mildly symptomatic mother. So we actually performed a Western blot on muscle biopsy from the mother, and you could see here that she actually had a reduced expression level of the ryanodine receptor. Once we had the mouse made, we actually went to see if the frameshift mutation was expressed, and you could see here that actually the exon 36 mutant allele is never expressed, and is the transcript is degraded by nonsense-mediated decay. Then our exon 91 missense mouse carries the A4329D mutation inherited from the completely asymptomatic father, who carries a wild type allele as well. And then we have the child who inherited the frameshift mutation from the mother, the missense mutation from the father, and actually, which means that this child has one allele only, which is encoding a homozygous exon 91 mutation. So we made an additional mouse model, which was the homozygous 91 mutant mouse. So we wanted to characterize the functional properties, both in vivo, ex vivo, and biochemical and physiological of this mouse model. The person who was in charge of these experiments was a postdoc in our lab, Moran El-Bas. So as you can see here, when the pups were born, actually, they were all indistinguishable, irrespective of what genotype they carried. There was no visible effect. But as you could see on this slide at the growth curve, although they started the same, starting at 3 weeks of age, you could see that the wild type, the exon 36 heterozygous, the exon 91 heterozygous, they're all the same weight, but the exon 36, exon 91 compound heterozygous mouse actually is smaller. And you could see this better on this picture. This is the wild type, and this is a double knock-in mouse. And the growth curves for both male and female mice show that the double knock-in mice are significantly smaller. We wanted to see if this was also caused by decreased food intake. And you can see here that actually the double hetero mice eat significantly less than their wild type littermates. So it's difficult to know whether they're eating less because their facial masticatory muscles are weaker, or they have more problems reaching the food. But part of the smaller phenotype may also be in part due to the fact that they're ingesting less food. So let's look at the physiological characteristics in vivo and ex vivo of these different mouse models that we made. So we allowed the mice to run on the voluntary running wheel. And as you can see from this graph, the wild type mice love running, and they actually, in approximately 3 weeks, they can run approximately 180 kilometers. They love jumping on the wheel. While the double hetero mice, you can see here, they don't even run half the amount, not even half the distance. So clearly they have something wrong with their muscles. Not only do they run less, but they also run much slower. So this is the speed at which the mean cruise speed of the wild type mice. And you could see that the double knock-in mice run at a much slower pace. If we look here on the side, what happens to the exon 36 heterozygous mice, the wild type, again, run approximately 180 kilometers, and the exon 36 mice run a little bit significantly less, certainly not to the same extent as the double knock-in mice. And the exon 91 mice, on the other hand, the wild type and the heterozygous exon 91 run the same amount, but the homozygous 91 also run significantly less than their wild type littermates. So then we performed in ex vivo experiments on isolated muscles, and we determined the mechanical properties of the isolated muscles. So it's clear from these twitch stimulations or tetanic stimulations of the EDL muscle or the soleus muscles that our double hetero mice develop muscles, develop less than 50% of the force compared to the wild type mice, both at the single twitch level or tetanic stimulation at all tetanic frequencies tested. This indicates that these muscles have severe impairment in their function. So the presence of the two mutations severely impacts how these muscles can contract. What about the exon 36 and heterozygous and exon 91? Well, this was quite surprising because the phenotype wasn't the same. So an exon 36 heterozygous mouse, only the EDLs contract significantly less than their wild type littermates, while the soleus muscles develop the similar amount of force. While tetanic stimulation, again, the EDL were affected much more strongly. If we look at the exon 91, on the other hand, on the homozygous exon 91 mice, the EDL muscles contract similar to the wild type counterpart, but the soleus muscles were weaker. So this is an intriguing observation because depending on the genotype of the mouse, where the mutant and which allele is missing, so either one allele is completely missing, we'll have an EDL which is more affected. And if both alleles are affected with a mutant channel, a homozygous mutant channel, in this case, only the slow twitch muscles are affected. We also performed, sorry, I have to move these because I can't see everything properly. So we also performed isolated single fibers from the mice and stimulated them electrically, either as a twitch stimulation or tetanic stimulation. And we isolated single fibers from soleus muscles, EDL muscles, or FTBs. And you could see that the red trace shows you the heterozygous, the double hetero mice, and you could see that all muscle fibers, in all muscle fibers, the peak calcium was achieved either by twitch or tetanic stimulation, was significantly lower in the double heterozygous mice. Now, interestingly, when we did this experiment with exon 36 mice, you could see here that again, it's the EDLs which are affected more severely than the soleus. And we did the same experiment with homozygous 91 mice. You could see here that the EDL are barely affected. And once again, the soleus muscles are affected. And this is quite intriguing because it indicates that once again, even though the reduced amount of ryanodine receptor we'll see subsequently is in both muscle types, clearly they're reacting differently depending on how the mutation is present. Then in collaboration with Rebecca in Oxford University, her group performed single channel recording on wild type or double hetero mutant channels. So she put them on, measured on single channel bilayer properties. And as you can see here, the voltage, the current voltage relationship of wild type and mutant channels is the same. However, if you look at panel A, you could see that this is the wild type channels. Depending on the calcium concentration, you push the channel to a more open probability. So the higher at low calcium channel, the low calcium concentration, the channel is closed. And as you increase the calcium concentration, the channel starts to open. And if you go very high calcium concentrations, you can see here that the channel tends to remain closed. Well, the double hetero mice channels behave differently. And you can see here that they have lost their calcium sensitivity, meaning that even if you change the calcium concentration, you don't have a change in the calcium open probability. So the double hetero channels have a reduced calcium sensitivity, and they also have a lower caffeine sensitivity. Finally, we performed ultrastructural analysis and biochemical experiments. And as you can see here, in the double hetero mice, the amount of ryanodine receptor in EDL and cilius muscles was always significantly reduced by more than 50%, while the histone deacetylase 4, which is a class 2 histone deacetylase, is significantly increased. There was, I don't have any of the pictures here, but there was a small atrophy of fast twitch muscles, but there was no change in the fiber type composition. And you can see here by electron microscopy that actually this is a wild type EDL muscle, which is very nicely arranged. And you have the Z bands, you can see the mitochondria and the calcium release units, while you could clearly see that these muscles here, they have lost all their amazing architecture, and you can see very, very few. You can see cores. These are mini cores, and you cannot identify very many mitochondria. Now, if you remember, I told you those are the characteristics of the fast twitch, EDL, and cilius muscles. But if you remember at the beginning, I said that patients with multi-mini-core disease or recessive mutations also have ophthalmoplegia. So this is the characteristics of these patients. And we were interested in knowing if our mouse model also had affected extraocular muscles. And if yes, why were these muscles also affected? So these studies were performed by Jan Eckert, a very talented PhD student in the lab. And the first thing that he actually did was measure the in vivo characteristics using this optokinetic reflex. So this is a behavioral test, and the mouse is placed in a cage, and there's these grids which are rotating, and as a reflex, he moves his ears. So you could see, however, that the wild type, the exon 91, exon 36, and double knock-in mice were all identical. And we're a bit surprised by this because we knew that the EDL and cilius muscles were actually affected. But this is a behavioral muscle, and what we realized subsequently is the reflex, although the mouse usually moves his eyes, in our case, he was moving his head. So then he still had the reflex, and this test is somewhat not valid for this measurement. So then Jan proceeded with measuring the force generated by these muscles during electrical stimulation. They're very difficult experiments because the muscles, as you can see here, are very tiny. So you could see that EDL, wild type exon 36 and exon 91 extraocular muscles all behave approximately the same way, but the double knock-in muscles fail to develop almost any force. You could see here a cumulative statistical analysis. So there's barely any force that's developed. This is very similar to what we were seeing in EDL and cilius muscles. We also showed that the weight, the length, and the cross-sectional areas of the extraocular muscles are the same in all the 4 genotypes. So clearly the extraocular muscles are also affected. And Jan also proceeded to measure the calcium transients in these isolated fibers from extraocular muscles. This is not an easy task because the extraocular muscle, he had to isolate single fibers. They're very small, very fragile. And you can see here a picture of an extraocular muscle fiber loaded with the calcium indicator MAG-Thul. He succeeded in electrically stimulating the fibers from the extraocular muscle. And once again, the double hetero mice are red. You could see here a superimposition of the traces. The double hetero mice, the calcium transient is approximately 50% smaller than that of wild type, while the kinetics aren't so affected. So once again, as we saw in the EDL and the cilius, we have smaller calcium transients in the presence of the double hetero mice. We then proceeded to do biochemical and ultrastructural analysis of the extraocular muscles. And this is where the interesting results we obtained. So we performed both Western blot analysis as well as mass spec. And it needs to be said that extraocular muscles are actually a combination. They seem to be a hybrid between cardiac muscles and skeletal muscles. So they will express RYR1, but then CAV 1.1, as well as CAV 1.2. They also express the beta 1 subunit. They express calsequestrin 1 and calsequestrin 2, circa 1 and circa 2, etc. And as you can see here, as far as extraocular muscles, and differently from what we saw in the EDL and Cilius, that both the ryanodine receptor and the DHP receptor seem to be present at reduced amounts, while calsequestrin 1 and calsequestrin 2 are present in higher levels. Now, another thing that we noticed, or Jan noticed, we had asked him to perform a myosin heavy chain gel just to see whether you could see a difference in the expression levels of myosin heavy chain. And just seeing this plot in this gel told us that the bands seem to be different. So at a better investigation, more detailed investigation by Western blot, Jan noticed that in the presence of the two mutations, you could see here that there was a band missing completely, and this is myosin heavy chain 13, or the extraocular muscle specific isoform of myosin heavy chain. This is a myosin heavy chain, which is called the superfast myosin heavy chain, which apparently gives the fastest contracture times to skeletal muscles. So there's a rearrangement actually as a consequence of the presence of these two mutations. There's also a downstream effect in the expression of the different myosin heavy chain, which are expressed in extraocular muscles. You could see also the EM microscopy. These were performed actually in Basel, and you could see the wild type mice. The structure of the extraocular muscles has a very nice architecture. You could see mitochondria, which are adjacent to the calcium release units, while the double knock-in, you could clearly see that there's no structure. The number of mitochondria are significantly reduced. The calcium release units, their number is reduced. We saw that there were more dyads present than triads. Dyads are never usually present in skeletal muscles or a characteristic of cardiac muscles. There was Z-line streaming, and there was a decreased number of mitochondria. Now, we thought this was very interesting because how can the mutations also affect the expression of the myosin heavy chain isoform? And two, what happens in the different genotypes? So we went to investigate because only in compound heterozygous mutants, or usually, that have expression of only one allele is extraocular muscle function impaired. So what Jan did, he performed Western blots on wild type, exon 36, exon 91, homozygous 91, and double knock-in mice. And he went to see if the extraocular muscle myosin heavy chain 13 was present. So you can clearly see here that in wild type and the heterozygous mice, they all express more or less the same amount of myosin heavy chain 13, but the homozygous mice actually expresses less. So 50% of the myosin heavy chain 13, but in the double knock-in mice, this is actually missing. So clearly there's a downstream effect caused by alteration of muscle structure or development. Further differences, we also did mass spec analysis of the extraocular muscles from the wild type and the double hetero mice. And you can see that there was approximately 1,000 proteins which were expressed differently within these two genotypes. And this tells you that there's downstream of the ryanodine receptor calcium signaling, there are many genes which are controlled by proper calcium signaling. And if the proper calcium signaling doesn't happen, you have misexpression of many genes. So some of these genes encode for proteins involved in excitation, contraction, coupling. Some of these genes encode for proteins involved in myosin heavy chain expression. And some of these genes are involved in mitochondria, oxidative phosphorylation, or mitochondrial proteins. And we know from the electron microscopy that the mitochondria are diminished in number in extraocular muscles from the double hetero mice. So this brings me to the conclusion and to the outlooks. We have created a transgenic mouse model knocked in for the compound heterozygous mouse mutation that recapitulates closely the phenotype of the patients with recessive mutations. In the exon 36, which carries the frameshift mutations, is not expressed. Therefore, only homozygous mutant channels are present, but to a degree of 50% or less. And these double knock-in mice are normal at birth, but then after 3 weeks of age, you can clearly see basically genotype just by looking at them, because they're significantly smaller. They run substantially less on the voluntary running wheel, and their mechanical properties and the calcium transients developed by the single fibers or their whole mounted muscles. You can clearly see that these muscles are very weak, and that the EM recapitulates the presence of small cores and disarray of the Z line. We also showed that the extraocular muscle function is significantly affected. They display reduced electrical evoked calcium transient, altered content of extra ECC coupling proteins, disorganization of the ultrastructure, and importantly, the absence of the extraocular muscle-specific myosin heavy chain 13 isoform. And finally, at the end, this is my acknowledgement slide. Of course, all these studies were done also thanks to Francesco Torzato. He actually developed mouse models in Basel. He designed them together with Pavel Peltsar. Most experiments were performed at the 2 postdocs in the lab, Alexis and Moran. Maya had started defining the extraocular muscle calcium regulation, and Jan performed all the experiments on the extraocular muscle fibers from the knock-in mice. Simona Boncompagni did the ultrastructure, and Rebecca Bassan from Oxford and her group performed the single channel recording. And of course, none of this could have been possible without our supporters, the patients, of course, and our funding agencies. Thank you all very much for your attention, and I'll be glad to answer any questions. Thank you.